Thank you for the big crowd that's here. Remember, when we have to applaud, you have to do it harder. Okay, fill the room. Okay. It's an honor for me to introduce to you Adrienne Edkins. Adrienne is going to tell us a little bit more about mycobacterial proteostasis. I'm not a biologist, as a drug target in tuberculosis.
Thank you very much. So thank you very much for the invitation. It's wonderful to be here to be able to share our work with you. So as you can tell from my title, I'm not a computational scientist. I'm a biochemist or a cell biologist dabbling in some of the computational modeling to help us to really understand biological problems. Next slide, please. Next slide, please. So to introduce us, I'm a SARCHI chair, South African Research Chair at Rhodes University. We're in the Department of Biochemistry. And essentially, we're based in the Eastern Cape. We're a fairly large group, but the work that I will talk to you about today is predominantly the work of a PhD student, Ronald Tanui. Next slide, please. So essentially, my group is interested in the concept of proteostasis. And that is essentially all of the collective processes that govern the production, stability, and turnover of proteins in the cell. And if you consider that proteins are by far the most predominant or abundant macromolecules in the cell, they're responsible for the majority of enzymatic and structural reactions. And so they form a very important class that needs to be maintained optimally for cellular health. And essentially, the group has three main interests. So we have a fundamental interest in understanding the mechanisms of these proteins. We then try to understand how proteostasis is dysregulated in the context of disease. And here we cut across multiple diseases, from non-communicable disease like cancer to infectious diseases caused by bacteria, parasites, and viruses. And then out of those two fundamental and more sort of applied research focuses, we then try to translate that information into either new drug targets or into identification of inhibitors that we can then use for this sort of next generation of therapy. Next slide, please. So proteostasis is really critical for cell survival, mainly because proteins are produced in a linear form. So they're translated by the ribosome and they come out as beads on a string, for example. But that's not the final functional form. They have to fold into a three-dimensional structure, and then they have to be regulated in a temporal fashion to make sure that they are expressed or turned over at appropriate times. And what we refer to as proteostasis is when the folding need, or the ability to fold and maintain those proteins, equals the capacity required by the cell. Next slide, please. However, every cell doesn't live in perfect homeostasis. It has to it does undergo stress at some point. And so at this point, we have a conserved system called the stress response that becomes activated in times of stress, or stress that particularly challenges protein structure and function. And in most cases, this is very well adapted. It produces a class of additional proteins called chaperones, and their function is to really help these unfolded or aggregated proteins to either reassemble correctly or to be degraded. Next slide, please. But in the context of disease, diseases related to aging or diseases related to infection, what we have is a scenario where folding need outstrips capacity, and so the cells can no longer cope. And essentially, that's when targeting or inhibiting components of the stress response becomes a therapeutic strategy. Next slide, please. So we're looking at this in various contexts, but today I'm going to talk to you a little bit about it as a target in tuberculosis. So tuberculosis is an infectious disease of the lungs. It's the leading cause of disease from infection worldwide. And it's caused by a bacterium known as Mycobacterium tuberculosis. And despite there being a number of antibiotics available for therapeutic treatment of tuberculosis, the treatment course is very long. And so that often leads to compliance issues, which then directly feeds into the development of drug resistance. And drug resistance is driven by mutations or changes in the sequence of proteins targeted by these antibiotics. Next slide, please. And so mycobacterium itself has a proteostasis machinery. The one that we are interested in particular is known as the DNAK system. And essentially, it's an essential class of proteins that regulate the function of critical cellular proteins. So we refer to these as clients. And these client proteins are the ones that are then required for survival or resistance to the antibiotic therapy. So even though the proteostasis machinery is regulatory, its functions or effects are mediated through these client proteins, which are either stabilized or destabilized. Next slide, please. So we started out this work doing some proteomics analysis, where we looked at proteins expressed in the lysates of bacteria in which we had either knocked down DNAK or which we had normal DNAK levels. And what we can see is that amongst these, so what we show here is a comparison, I hope you can read that, but a comparison of the proteins that were dysregulated upon inhibiting the proteostasis machinery with all those proteins that are required for an essential survival of the Mycobacterium tuberculosis bacterium. And what we find is that just under half of the essential genes required for the survival of Mycobacterium tuberculosis are deregulated when you deplete this chaperone system. And in particular, a number of them are related or targets of particular antibiotics. And so the story I want to tell you today is about a protein called RPOB, which is a member of the RNA polymerase. And we find that in our lysates, RPOB levels are deregulated and they're slightly upregulated with DNAK depletion. Next slide, please. So RPOB is part of a multi-protein complex that forms the RNA polymerase, and it is the target of the drug rifampicin, which is one of the first-line drugs for TB treatment. And rifampicin resistance arises due to mutations in the RPOB molecule. So the first question that we wanted to ask is, well, is DNAK involved in, or the chaperone machinery involved in promoting resistance to rifampicin? That would then give us a link to the changes in RPOB levels when DNAK levels change. Next slide, please. And what we can see here, if you look at these slides, we are essentially looking for the presence of bacteria that can grow in elevated levels of the antibiotic. And so a high number of bacteria correlates with a high development of frequency of resistance. And so we can see that when you introduce higher levels of Mycobacterium tuberculosis, you drive resistance to rifampicin. And so we wanted to say, okay, well, this suggests a relationship between rifampicin and the chaperone machinery. And does DNAK or that chaperone family influence RPOB activity? Next slide, please. And what we can see here, so here is essentially an assay looking at RPOB activity in vitro. And we can see that as we stress the system, so when we place the system under stress, we sequentially lose RPOB activity. So we lose the ability to make an RNA copy from a DNA template. But when we include the Mycobacterium tuberculosis, that is sustained. And so this is suggesting to us that, yes, DNAK is involved in driving resistance, and it's involved in directly stabilizing this and counteracting stress encountered by RPOB. And so then that brings us to the question, well, all of these studies are done in the context of the wild type protein. So do resistance mutations that arise due to rifampicin resistance, how do these then influence RPOB stability and its association with DNAK? Next slide, please. Okay, and so obviously the fact that we have so many resistant TB strains at the moment means that there are very well characterized mutations associated with rifampicin resistance. And you can see that single point mutations, either alone or in combination, are associated with substantial acquisition of rifampicin resistance. And so we started looking at these proteins, these amino acids. If you locate these, you find in the context of the full-length protein, the majority of them map to a small region known as the RRDR, so the rifampicin resistance defining region, within a central cavity where rifampicin binds. And so at this point, we were interested now to try and understand if we could use simulations to see whether there are changes in the protein structure that we could then relate to the acquisition of this resistance. Next slide, please. And so we started out doing some static docking. So this is 5 different algorithms that were used to look at or predict the effect on stability of these single point mutations or combinations of those mutations in RPOB. And what we found was that it was really not sufficient for our purposes, and we couldn't get good agreement between these different models. And this is really when the point in the project at which we turned to the ChPC. Next slide, please. So essentially, we wanted to say, well, can we now take from the static docking, can we rather use molecular dynamics to understand how these mutations change the protein and whether this is going to influence its stability and predict a reliance, for example, on the proteostasis machinery. So this was the work that was done by Ronald, and essentially what we've used so far in terms of computing hours, about 800,000 hours on this particular project. Each of our runs about 48 hours, involving 240 cores, each with 10 nodes. We started out with optimization runs at 25 and 40 nanoseconds, but we're not able to get the system to reach equilibrium within that time. And so we essentially ended up on 100 nanosecond runs, which gave us at least a saturation or an equilibrium for the system. We did this in triplicate for 7 different RPOB variants, generating using substantial storage space. And obviously this project is still ongoing, and I'll mention the work that we want to do beyond what we've done so far. Okay, so next slide, please. All right, so essentially we use an MD simulation analysis pipeline using GROMACS. So this is an established MD simulation pathway in the field. So it essentially starts with sequence retrieval, introduction of the mutations, the point mutations at specific points and alignments, modeling of the 3-dimensional structures, followed by alignments of the wild type and the mutant proteins. And then essentially the next set of phases basically create a box that recapitulates as closely as possible the environment within the cell. And that then allows us to run the molecular dynamic simulations under conditions that would be as close as possible to what the protein itself would anticipate within the bacterium. And then once we have the simulations, we can do the post-analysis, including looking at global and local changes within the protein structures and other sorts of analyses. And obviously, at this point, we're still doing many of these analyses, but I'll talk about what we found with respect to the first 2 now. Next slide, please. Okay, so this is showing the RMSD value, and you can see that the wild type protein, which is the native protein that has not acquired any resistance, has showing its trend here, and we can see the system saturates after about 60 nanoseconds. And what we see is that with the exception of this S450L mutation, all of the proteins show increased higher RMSD values, indicating conformational flexibility relative to the wild type. And this is a global increase in conformational flexibility in these proteins. Next slide, please. In addition to that, we can link it with, sorry, that should be RMSF at the top there. Forgive me, that should be RMSF, looking at positional residue flexibility. So how flexible are individual residues at specific points along that protein sequence? So you can see it's a very large protein, which is one of the reasons why we had to use the computing power from ChPC. And what you can see is as you track along that protein sequence, we have regions where there is very little deviation between the wild type and the mutants. And then we have regions where we see large fluctuations, in particular here in the C-terminal region of the protein. and another region here towards the N-terminus, and then in particular in this region, which defines that rifampicin resistance domain. Next slide, please. And obviously we did this in triplicate, so the average values again just reinforce what we saw in the files or the graphs there, which is most of these proteins show values that suggest that they become conformationally more flexible, and that the residues themselves within the protein become more flexible and less rigid compared to the wild type structure. Next slide, please. So we then turn to a set of in vitro assays that we could do to compare to what we had seen with the in silico predictions. And so we could test the ability first of these proteins to interact directly with DNAK. And then secondly, we could look at their stability, their thermal stability. And this is essentially a process where you unfold a protein in a test tube and you monitor the temperature at which it transitions. And that gives you the TM. And obviously a higher temperature of TM indicates a more stable protein. And so we can see that all of the proteins will interact with DNAK, and although there are some differences in the affinity, those differences are rather minor. However, we see significant changes in the TM. So a TM of 2 degrees is considered highly significant. We see that the wild type protein has a melting temperature of 38 degrees, which indicates it's a fairly unstable protein. And all of these variants are shifted to higher temperatures, suggesting in this case that we actually have an acquisition of a protein that is more stable in its folded form. Next slide, please. And we can also then compare folded protein stability to the concept of aggregation, okay, which is when proteins start to lose their secondary structure and they aggregate into insoluble factors. So what we do here again is you incubate the proteins in solution for a period of time, and then you can monitor the transition from an aggregated to a soluble to an aggregated state. And what we've done here is just normalize that at 100% for the wild type, okay? So if you just look at the black bars, you can see that all of the mutants show enhanced aggregation above the wild type, okay? However, they can be successfully captured by DNAK. So these shaded bars here indicate the aggregation when you're in the presence of DNAK. So these proteins can be successfully captured and their aggregation suppressed to an equivalent level of the wild type protein. So even though they're more aggregation prone, they are still very efficiently suppressed from aggregation by DNAK. And this implies a better ability of the system to suppress aggregation in disease-causing variants of the proteins that drive drug resistance. And so this is very promising in terms of developing new therapeutic strategies targeting DNAK and the chaperone machinery as treatments either alone or in combination with existing antibiotics. Next slide. So in summary, we see from the in silico analysis that we observe with most of these mutations, changes in protein conformation and increased residue flexibility. But this seems to be associated with actually an acquisition or an increase in stability in the folded form. However, the proteins are still more aggregation prone. And despite the fact that they show this phenotype, we have equivalent or better stabilization by the proteostasis machinery, suggesting that they may be more sensitive to inhibition, resulting in aggregation and then turnover. So we've obviously only started this analysis. We're still doing hydrogen bonding and salt bridge analysis and some additional data post-analysis on the simulations that we have now. But we'd also really like to do MD simulations of the complexes, now that we have some evidence to suggest that there may be a different regulation at that level. And then we're obviously linking this into studies looking at the pharmacological and genetic inhibition of DNAK as a treatment, not only for TB, but in particular for drug-resistant TB. Next slide, please. Okay, so it just remains for me to thank you for your attention and just to acknowledge collaborators who have worked with us on the project. So Garth Abrams is TB expert at Rhodes University, Clint Field at UCT. Alex did the mass spectrometry work for us. And obviously we are very grateful to the ChPC for giving us access to their resources that have allowed us to really start now to transition and get insight into what's happening at the protein level in terms of our work. And then also just to say that Ronald and Shishala were the main drivers behind this work. And somebody said yesterday, one of the student presenters said that they were very glad that their supervisor was in the audience because they could answer the tough questions. So I'm going to end by saying I'm very glad that Ronald is in the audience and he will be asking any of the tough questions. Thank you very much.
Thank you for a very interesting presentation. Even I understood it. So thank you very much. Any questions?
Yeah, thanks, Adrienne, for the talk. Just a quick question. Maybe I missed it, but do you make use of the GPU cluster? Why not?
No, we haven't used the GPU cluster. All of this was run on the CPU, and we would very much like to use the GPU cluster. So if that is an option, and I think we would have to do it if we wanted to do the complex analysis.
Yeah, most certainly for the molecular, for the MD runs, I would suggest let's talk about the GPU cluster, because it seems like you are running out of capacity a bit on, if you're already on 10 nodes on the CPU cluster. Certainly you might be surprised with the performance increase that you will see.
Well, that would be wonderful. Thank you.
Good.
Any other questions?
Hello, hi. Thank you so much for your talk. I was just wondering if you could elaborate a little bit further of how you calculated the changes in the structure as a result of the point mutations that you had.
What do you mean? You mean in terms of the RMSD and the RMSF analyses?
No. So like, how did you? Because I'm presuming that you have a initial structure sourced from a crystal structure, something like that. I'm just wondering how you then got from that structure to the structure as a result of the point mutations.
Okay, so we did that with molecular modeling. So we have a modeling pipeline that we use. So the structure that I showed, we would have the wild type sequence and then use that essentially as a template in Swiss Modeler. And we have a pipeline called Primo that was actually developed at Rhodes University. And so we essentially follow that process through the introduction of the mutations, the modeling of the variants, obviously the model validation, before we then input that into the molecular dynamics.
Thank you very much.
Yes, thank you very much. So I have a question from my small physics brain, but this idea of the mutations being more stable, but also more flexible. So the RMSD values being larger, with the melting temperature being larger. Can you maybe explain that?
Yes, so on the face value, those two seem contradictory. And I guess the way that I would conceptualize that is perhaps, so the sort of dogma is a single sequence, a single stable structure, obviously. And the way that I conceptualize the fact that these residues are perhaps moving more and are more flexible, with the consequences then for essentially the folding, although we measure folding, it's unfolding, it's a proxy for folding. So I would suggest that perhaps because those residues are slightly more flexible, they are then more tolerant of small changes, and they can perhaps have a greater propensity to resist the unfolding in that sense. So that's the as far as I've got in terms of conceptualizing how that happens. But I would agree with you that it seems contradictory.
let's thank Andrea.
Thank you.